Hello, everyone, and welcome to Concordia University's Fourth Space. Thank you for joining us for today's event, Life But Not Alive, Synthetic Cells. To help situate you, we are streaming to YouTube live from Fourth Space, which is located on unceded Indigenous lands in Tiohtià:ke, Montreal. Here at Fourth Space, we work with our university community to mobilize knowledge by co-creating daily activities to examine research questions and projects in development here at the university. We are running today's event as a live stream Zoom meeting, so for those of you joining us on Zoom, we welcome your comments and questions with a raised hand or via the chat. And for those of you in the space, if you'd like to participate, just let us know by raising a hand and we'll be sure to get a microphone to you. With that, it is my pleasure to hand it over to the Manager of Synthetic Biology Applications CREATE program at Concordia University, Orly Weinberg. Orly, welcome.
Thank you very much. Glad we could be here at Fourth Space today and share this very interesting talk with you. We have with us guest speaker, Dr. Kate Adamala from the University of Minnesota. Kate is the McKnight Presidential Fellow Associate Professor at the University of Minnesota. Her research focuses on synthetic cell engineering with the aim of understanding chemical principles of biology using artificial cells to create new tools for bioengineering, for medicine, and in order to do foundational research. The interests of her lab span questions from the origin and earliest evolutions of life, using synthetic biology to do all sorts of interesting things, colonize space, looking at the future of biotechnology and medicine. Kate is a co-founder of the synthetic cell therapeutics startup company, SynLife. She is also co-founder and coordinator of an international synthetic cell engineering consortium called Build a Cell Network. And she's a polymath fellow at the Geneva Center for Security Policy. So she's involved in all sorts of interesting discussions about how we regulate policy for synthetic biology. It's my pleasure today to bring on Dr. Kate Adamala. Kate?
Thank you so much, Orly. Thanks everyone. Thanks for being here first thing in the morning. It's my pleasure to talk to you. I want to move around so I can see you and not the lights. Okay, that works great. Okay, so what I want to talk to you about is what are synthetic cells, why we're making them, what's the point of this whole field in general. So when you think about biology in general, can we get the slides up so people, oh yeah, awesome, thank you so much, awesome. So this is biology. Biology is awesome. Biology is incredibly diverse. When you think about it, biology probably has done everything that you can think of already. So what's the point of making synthetic cells? What's the point of making cells from scratch if we have this incredible diversity to study? What might ask? And that's a valid question. So when you think about it, all of biology, if you've done chemistry, if you studied chemistry, you realize that all of biology really is on the chemical level incredibly boring. And I know that probably doesn't sit well with biologists to say that, but when you think about the roots of life, when you think about the chemistry, all of biology has been done on a sample size that equals 1, because we only have one type of modern terrestrial life. So that's the life that we know of. So all of the studies of biology, and most importantly, all of the engineering of biology has been done on that single sample size that is the modern terrestrial cell. And on a biochemical level, it's all the same. All cells use the same 22 proteinogenic amino acids. All cells use the same nucleobases, the same genetic code, with very few exceptions in some weird organisms. So really, when you look at the tree of life, this incredible diversity of life, what you really should see as a chemist is a single architecture, a single type of chemistry. And most importantly, we don't even know what that single architecture does, how it's built. So we don't have a complete list of proteins and small molecules of any living cell, despite all those years of studying it. We've made a cell from scratch. We've made a cell with a completely synthetic genome, so we can tell you exactly what is the sequence of a genome of at least a minimal living cell, but we still cannot tell you what each of those genes is doing. So essentially, life is a black box. If you look at any biological cell, all the work that's been done so far in biology still hasn't really explained to us how even a simplest living cell works. So what kind of engineers are we really? I mean, we call ourselves bioengineers, but it's really kind of like poking something with a stick and hoping it's going to work, because we don't know exactly the mechanisms inside it. And so that's the rationale behind the field that I represent. We say that if we can't really understand something, we can't engineer it. And there is a terribly overused quote from a physicist, Richard Feynman, who said, "If I cannot build it, I don't understand it." It's a cheesy quote because everyone uses it in this field, but it really makes sense. It really is very relevant today. If we cannot build a living cell from scratch, then we cannot say we truly understand what life is. And most importantly, we cannot engineer it. We can't really engineer it with like all of the precision that is required of engineering if we can't say we know exactly where every molecule goes. So that's what we're doing. We're engineering life from scratch, from non-living components. And we're hoping that once we make a living cell from non-living components, we'll be able to truly understand it. Not just truly understand life as just a physical phenomena, what is life, those big philosophical questions, but also practical questions. How can we make biology work for us? How can we engineer biology so it does what we really need it to do? So what is that we're doing? We're making a synthetic cell. So a cell has a membrane. That's kind of the business model of a cell. When the cells were first invented, the man who first saw cells under the microscope, he called them cells because they looked like the cells that monks live in. So a cell has to have this compartment, a membrane. And inside that membrane is made out of the same lipids as all of your membranes. So when I make a synthetic cell in my lab, I use the very same lipids that you use in your own membranes. So each of those lipids has kind of two parts. There is the tail of a lipid. So the tail is what really wants to stick together, go inside this kind of inner part of the membrane. And there's the head group that sticks out to the world, to the water environment. And that's what really kind of represents the surface of the cell, and that's how we can engineer the interactions between the cell and the environment. Now, that whole point of the compartment is to separate what's inside of the cell from what's outside, the external world. That's how life works. Life separates itself from the external world. Life has individuality. So that's the whole point of our compartment. But a compartment doesn't really work well unless you can transport things across it. So that's why we put membrane pores, membrane channels into our membranes. So we put those channels in membranes that let us bring things from the outside and eliminate waste from the inside, just like natural cells do. Except I actually understand what my pores do because I put them in there. I engineered those pores. Now the really fun part is what's inside a cell. And so what's inside the cell is these enzymatic reactions that are supposed to mimic the inside of a normal living cell, so inside of the cytoplasm. And those enzymatic reactions is where the real, I don't want to call it magic because we pretend we don't do magic, we do actual honest science, but that's where the magic really happens. That's where we do the engineering. That's the part of the cell on which we do the engineering. And so what's inside that cell? So it can just be simple reactions. This is a video from a postdoc, Theodore, in my lab, who works on membraneless and membrane microfluidics. And so these are just millions of synthetic cells that we generate from this little microfluidic device that we can make in those systems that are really well controlled, just to show you how these guys look like under the microscope. But what's the point of it is that you have that membrane with the pores. So this is how every cell on Earth right now works. These are how the cells that we build work. And then inside that is that interesting translation system. So what it does is we have a genome. All life has DNA, all life has genomes. I told you just a couple of minutes ago that we want to kind of move away from natural biochemistry. We want to build life in ways that natural life doesn't do it. But for now, we're stuck to the building blocks that were available to us. So that's DNA. So all of synthetic cells that we've made so far have DNA genome, just like your own genomes, except much smaller. How much smaller? So a human has, about 100,000, maybe that there's a debate of how many genes, about 100,000 sequences that actually get used. Out of that, about 20,000 of them are protein coding sequences. So that's a good sized genome. How does that compare to other organisms? A bacteria. This is a representative genome for E. coli. It's about half the size in protein coding genes. There is a debate on how many really there is. This is, by the way, this is a log scale. So this is a logarithmic scale. So you can see it's not a linear scale like you're used to. This is log scale like we use in science to emphasize those small differences. So this looks like a pretty small difference between a human genome and a bacteria genome. We're not that different. Just to put that in perspective, I told you we made a synthetic genome of a living cell. We made basically a cell with a completely chemically synthesized genome. The size of that genome is about an order of magnitude smaller than the size of a human genome. So that mycoplasma cell made first by Craig Venter about 15 years ago has a completely synthetic, chemically synthesized genome. Now, where do I place on that scale? So that's the reason why I use the log scale, because if it was a linear scale, you wouldn't see anything. We are right there on the bottom. The synthetic cells that we build have genomes that have between 10 and 100 genes. Now, that sounds like not much. That sounds like it's a really wimpy genome, and true it is. But if people have done math and figured out that to make a independently living cell with all the components that most of us would agree are necessary for life, you need a genome that's about 120 kilobase pairs in size. We are now in synthetic cells at about 90 kilobase pairs. So we're getting there. We're getting to the order of magnitude of what's needed to really make a synthetic cell with a completely synthetic genome. We're still early at it. I mean, we haven't really made life from scratch yet, but we're getting there. Just to show you that we're not playing with 1 or 2 genes. We're actually building genomes already. Okay, so this is our DNA, but DNA by itself really does nothing. It just sits there. In order to make use of DNA, you need to express it. And so what we do is we express our DNA in a translation system. So you used the word translation before. It's the process of making proteins. So you have a genome and you want to express that genome, you really make use of that genome, you need to translate it. So that's the process of making proteins, and we can reconstitute that process entirely in vitro, so in a synthetic system. So when I make proteins, I don't need a living cell to make proteins. I just take my genome and make proteins out of it. So all of those components that you need to make proteins are in a synthetic cell, are made and allow us to basically take the genome, express it into the proteins, and then those proteins have function. What are those functions that's really the fun part. Okay, so you can think of a synthetic cell kind of in a way like a jigsaw puzzle. Normally when you do biological engineering, if you work with bacteria, most of the time you work with bacterial genes because that's all that your bacteria can express. If you work with a human cell, most of what you're expressing are human cells. What we are able to do in a synthetic cell is express genes that come from a lot of different sources. So we can basically put almost a zombie cell together. We put elements that come from a lot of different organisms. So you can, for example, take a genome of a plant, add some bacterial genes to it, add some enzymes from a fungi or a jellyfish. Actually, we use jellyfish because that's one of the most common reporter proteins that we use. And then you can put all of that together. You can add some human genes, so you can basically mix and match elements. So that's getting to be real engineering. We can actually take elements from different sources, put them together, and create something new, create new functionalities. And that's what really helps us go beyond natural biology, even in this very still biologically inspired system. So how does it look like under the microscope? So this is a representative image from one of the experiments done in my lab. It's not much to look at, it's not much to write home about, but this is a cell-like structure that was made completely from non-living components. So what you see here is just 3 images. This is the inside of the cell, so this is the protein expression. By the way, that green protein, that's the jellyfish protein. This is called green fluorescent protein, which literally is a protein that's green and fluoresces in green. Not very creative name, but that protein did come out of a jellyfish. It's one of the favorite proteins of synthetic biologists. So that shows us that there is a cytoplasm, there's protein expression inside our synthetic cell. We can label the genome. As you can see, the genome is very evenly distributed inside the cell. We can label that genome with a genome stain. And then there's a membrane. I don't know if you can see it. I'm blinded by the lights. If you can see a red circle there, if you don't, trust me, it's there. It's the red membrane stain. So that's the membrane of the synthetic cell. So that's how it looks like. It's not a little green alien that waves its hand to you from under the microscope, but it's, we're proud of it because we made it with our own hands. We made that cell-like structure literally from non-living components. Okay, so this is what we want to do. We want to make life from non-living components. How do we do that? There are two different ways that people go about it. One is the right way. That's the way I use. That's taking non-living components and putting them into a cell. Right now, it's not yet living. So we put together those elements of cells, we make a cell-like structure. Some physicists argued with me that what I make is already living because it satisfies some arbitrary definition of life that they have, but they're physicists, they always have arbitrary definitions. I don't think it's yet living. To me, to be living, you need to self-replicate and evolve. So our cells are not yet living. We do have a living cell, a minimal living cell. This is that cell I mentioned before. This is a cell with a completely synthetic genome. That cell has been made by minimizing a complex living bacteria. That cell is undeniably alive. It's small value of life. It's really wimpy organism, but it is living. But we still don't understand what every gene in that cell does, despite us making that genome from scratch. So the hope is that one day both of those approaches, we call them top-down and bottom-up. The bottom-up is taking non-living things, putting them together. Top-down is taking something complex and trying to simplify it. We're hoping that one day those approaches will meet in the middle and we'll actually be able to make a living cell that's completely understood, that we can tell you exactly where every molecule in that cell goes. So imagine it's a living system, but you know exactly where every molecule of it goes, so we can truly understand what life is finally. So we're hoping that one day that's going to happen. Okay, so that sounds pretty crazy, right? I just told you we want to make life from scratch. No one has ever done that before. I want to show you a quote from an article that was published over 100 years ago in the New York Times. So the New York Times is a very respected publication. It was even more respected 100 years ago. Someone published an article on the feasibility of building a flying machine heavier than air. So this is, the year is 1903. We have balloons and we have the dirigibles, the blimps. They all fly because they're lighter than the surrounding air. That's the principle of a balloon. That's the principle of a blimp. So people have been speculating that it will be possible to build a machine that flies that's not lighter than air. So they call them heavier than air flying machines. And so someone published this very eloquent article saying this is absolutely nuts, impossible, don't even try. And so what happens when you tell scientists and engineers that something is impossible and we shouldn't try is we're just going to go and do it. And so 2 months after that article has been published, the Wright brothers flew their first airplane. And the reason why I love this quote, especially with the timing coinciding with the first machine-powered flight, is that it really shows you that what sounds like science fiction, what sounds like impossibility, or even you can have physical proof why it's impossible, and then it happens. At some point, people go and do it. And so that's kind of the whole principle behind our field is we say we're going to make life from scratch, and people are skeptical, people say it's not going to happen, but then we just go and do it. So once we do it, what are we actually going to do with it? What is it good for? So one of the things that it's really good for is to understand natural biology, to understand how biology works, understand every process in biology. And that's obviously going to be helpful when you try to make tools to engineer biology, but also when you try to make things like medicine and diagnostics tools. Because a lot of the problems we have, current medical problems, are not solvable with the tools we have right now as we know it. We've been putting money into biomedical R&D and less drugs is coming out. So we need kind of a new way of thinking about it. And we're hoping that if we have a synthetic cell that allows us to kind of change the way we think about making drugs. There are other cool things that we can do with it. We can make new types of materials. It can help us with space exploration. It can help us with understanding the beginnings of life on Earth. So literally how life started on Earth. And it can help us make things like new materials. It can help us make biocomputers. That's one of the things that my lab is working on, making biological computers. Again, science fiction. It's probably not going to happen anytime soon, but we're working on it, to making a computer that's made out of living cells. So all of those really cool applications are coming up from this technology. And the question obviously is, what is it good for you, for everyone in this room, for everyone who's listening to us online? What can you do with it? What's the point of doing it? That's not just some scientist pipetting in the lab far away from you, but what is it actually? How does that affect your everyday life? And so it does affect it. One of the ways in which it affected is because what you're doing in your everyday life right now is unsustainable if we're going to survive at least 100 more years as a civilization. And the reason for that is because right now all of our world relies on oil. And when you think of oil, you mostly think about energy. But there is so much more that comes from oil: plastics, drugs, everything around you, your clothes, the chair you're sitting on, the medicine you took this morning comes from petrochemicals. And last but not least, a lot of people don't realize that the reason we can feed the world right now is because of petrochemicals. Not because of transport or an oil used as gasoline, but because of fertilizers. We could not feed the current population of the world using traditional agriculture. The reason we can produce enough food for the current world population is because petrochemical-derived fertilizers. So without oil, and specifically without petrochemicals, we could not have the world as we know it right now. So we have a lot of alternative energy sources. We have the wind, solar, we have nuclear energy, but that's just energy sources. That's not the sources of the chemicals that we right now derive from oil. So just with the current technology for renewable energy sources, we could make electricity that can power cars, lights, that can heat our homes, but we don't have right now a source to replace oil as a source of chemicals in all those other applications. So we couldn't make fertilizers, we couldn't make clothes, computers, medicine. Anything that uses petrochemicals right now could not be made with those renewable energy sources. So we need this new technology, we need some new way of making all those chemicals. And one of the things that the world has in abundance is green stuff, plants, grass, prairie, whatever grows in every called. And so we're starting to learn how to use that, how to use what grows naturally in every environment and turn that into chemicals. And that's a process called biomanufacturing. So we can build those reactors, they're literally like chemical reactors, but they use green biomass as a feedstock. And inside those bioreactors, we can turn prairie, grass, whatever grows naturally in every environment into chemicals. And those chemicals can then be used to replace anything that otherwise right now we make from oil. And that includes making fertilizers for crops, making plastics, making everything that right now comes from fertilizer, that comes from petrochemicals. And so can we actually do that right now? Well, we think we can, but it's not yet done at scale. And one of the reasons why it's not yet done at scale is because natural bugs, so those bioreactors are filled right now mostly with bacteria. And bacteria are not dumb. They're not going to redirect their entire metabolism to make what you want them to make, in many cases toxic, petrochemical equivalent chemicals, because their first order of business is to replicate, to grow, to be healthy. And so that's why we are developing those new technologies. So what can we put inside that bioreactor? Right now, it's a living cell mostly, but we're starting to put synthetic cells inside those bioreactors. And some of the advantages of synthetic cells that we have using those synthetic cells over natural cells are really relevant, especially for building that bioindustry, for building that bioeconomy based on biomanufacturing. And so one of those big advantages is that it's safer to use synthetic cells. It's much faster to develop different applications, because when you want to really have a bioproduction that's agile, responsive to the needs of the industry, you need to not wait a year to make a new bacterial strain. You need to be able to retool your production within a week. That's much easier to do with a synthetic cell. And last but definitely not least, it's much cheaper to use synthetic cells. You can basically engineer that cell in a short amount of time. It's safer, and you have to spend less money to do it. So this is where the world is hopefully going. We're hoping that we'll be able to do this bioproduction at scale. But there's some other really cool applications of this technology. So one of the things you can do with a synthetic cell that you cannot do with a natural cell is that I can send you an entire ingredient list of a synthetic cell. So I can literally digitize my cell and send it to you, and you will recreate it in your lab if you have one. Hopefully one day people will be able to do that in much less resource demanding environments. And so that means we can send information rather than sending materials. So for example, if I have a collaborator in Europe, I don't have to send them physically my material. I can send them just the information about making a cell, and they can recreate that. So that literally is teleportation. We can send something electronically, and that gets recreated from the material on site. You cannot do that with living cells. So obviously that's, for my collaborator, for example, in the Netherlands, that's neat, that saves just a little bit of time because FedEx delivers there. What if we want to build synthetic cells that are used in environments where FedEx doesn't deliver? For example, we talk about manned long-term space missions. We talk about sending Elon Musk to Mars. Once that happens, we're going to have to address the needs that we don't know about in advance. So we want to make synthetic cell platforms that allow us to make drugs, to make nutrients, to make small molecules that are going to be needed, but we don't know in advance what's needed. So me in my lab on Earth can develop the technology that, for example, makes a cancer drug. Once the crew develops a need for that drug, we will send them information how to build that drug, and they'll be able to make that platform hopefully in something like 6 to 8 hours. So we send them information, they use the set of tools that we already gave them on the launch, and they'll be able to build that biomanufacturing platform. It's called biomanufacturing on demand because you can develop it as it's needed. It also has a lot of uses for applications on Earth in environments where you don't have a lot of resources. And actually, one of the greatest pioneers of the applications of those technologies for low resource environments on Earth in diagnostics is in Canada. He's in Toronto. Keith Pardee develops those diagnostics platforms for places where it's difficult to deploy modern medicine. So for example, he's done field trials for Zika virus detection in South America. And so you can do that on demand from those systems that are freeze-dried. Does it really work under those conditions? So we show that it does. This was one of the most fun projects that I've ever done in my lab. We made a payload for trials of that technology in orbit. So what I'm showing you here is a video of a launch of the first Sounding rocket with a synthetic cell payload. I had to get up at 3 a.m. to watch that launch, so now I'm forcing everyone to watch that video. But it's also, it's inherently cool. It's a sample that I prepared with my own hands. Now it's going into space. That was really cool for me, but also really cool for science because we did demonstrate that it works. The next stage of that project was sending a sample to the ISS, and we had an actual human astronaut pipette the sample, add water to our lyophilized sample, and produce, again, a reporter protein. So it does work under spaceflight conditions. So applications of that are not just to explore the universe with a human space flight, but also kind of a more esoteric but really cool applications like looking for life elsewhere. Because with synthetic cells, we can recreate the earliest evolution of life. So when you're wondering how did life start on Earth, right now we have no idea, we just know it happened. But if we can recreate that process in the lab, we not only will be able to understand how life started on Earth, but also looking for life elsewhere. We're right now preparing sample return missions from Mars. There's already a site picked where we're going to drill under the ice on Mars. Mars is probably contaminated by now with all the crap we crashed there, all the hardware that was probably contaminated with terrestrial bacteria. But there are places in the universe that we haven't contaminated yet. For example, Enceladus and Europa, there are 2 places in our own solar system that have liquid water. And if there's water, there's life. That's kind of the mantra that we say. I don't know if it's true because we haven't found life outside of Earth yet, but we're hoping that once we have a sample return mission from Europa, we'll be able to drill under the ice there, pick up a sample. What are we looking for? That's a very good question because we don't know how life could look like, a life that originated outside of Earth, because all we know is terrestrial life. All we know is how life looks on Earth right now. So the idea is that if we can use synthetic cells to recreate that prebiotic evolution in environments that are not Earth, for example, I can simulate the European ocean in my lab, and then we can say, okay, that's how the life would look like if it evolved there. So when we actually go there and pick up a sample, we'll be able to know what to look for. So that also helps with space exploration, not just with sample returns in a solar system, but looking for life elsewhere in the universe. So, is there life elsewhere in extraterrestrial solar systems? We know there is a lot of exoplanets that have the conditions that are similar to Earth. Can we look for life there? We can look for life there spectrophotometrically. So we can look at the spectrum of those planets, and we can say, are there molecules there that are hallmarks of life? So doing those origin of life experiments in the lab allow us to kind of understand how life could start elsewhere. Okay, so back from the galaxy, what if you get sick? Right now, if you get sick, you either get the small molecule drugs that we've been using for 100 years, or if you're really lucky, if you're really unlucky to get a rare disease, and then you get really lucky to go into a medical center that offers personalized medicine solutions, you will get a molecule that's been produced for a big patient population. So for hundreds or thousands of people that have a disease similar to you. So we can do that right now. We can produce those molecules in bacteria or other organisms. We can produce those complex, mostly peptide drugs, and we can give it to you. But every patient is different. You are different than another person that even has a disease that's called the same as your disease. But it's different because you are different. And that's the whole concept of personalized medicine, is that we should really make medicine for every single patient separately, because every patient is different. And it's really difficult to do that with a natural cell bioreactors. If you remember that a few minutes ago, I was comparing synthetic cells to natural cells. Synthetic cells are cheaper, easier to work with. So the idea is that we would like to develop a synthetic cell bioproduction platform where every patient gets their own little on-demand portable biofactory. And that portable factory will make drugs for every patient. So when you're sick, we're making drug for you, not a drug for 1,000 people that look sort of like you, and maybe that drug will work for you, maybe not. That's the idea. We're hoping that will be the future of medicine. So how do we get there? There's a lot of cool applications, but we're obviously not there yet. So the kind of idea is that we want to get those technologies out of the lab and into the clinic, into the companies that will actually start commercializing that technology and really bringing you those solutions, those applications. And so we do it as an international community. That's one of the things that I really love about my field, is that we're not just working in our own labs. We're not just collaborating with a friend in a different state, or like me collaborating with someone in Canada across one border. We really are an international community, because what we're trying to do, it's crazy making life from scratch. What we're trying to do is really not doable by a single community or even a single country. We're building this international community across the borders, and we genuinely are able to work across the borders, mostly because there's not many commercial applications yet, so there's not much money, no one hates each other yet. We still like each other, we're still friendly to each other. But we're hoping to preserve that as the applications develop. So I did the dumbest possible thing a junior PI can do. When I started my lab, the first year I started my lab 6 years ago, the first thing I did was to say, okay, there's this idea of building synthetic cells, we should do it internationally. Let's start an international community. This is the last thing you should do when you want to establish yourself as a researcher, when you want to start your lab. But it did work out, fortunately. There were other people that wanted to go in on it with me, and that's how Build a Cell was born. Build a Cell is an international community that coordinates creating synthetic cells. And one of the best things about our community is that we actually physically meet twice a year. So every 6 months, we have a Build a Cell workshop that meets in person, and we spend one day actually working. So these are workshops where people actually sit around and work. There's no talks. This is people get in the room, take out their laptops and work. And obviously we do work more frequently than every 6 months, but every 6 months we do connect with everyone in the community. So we actually know each other. People come to those workshops from Europe, from Asia, from all over. We actually get to meet each other, and so not just the PIs not just the professors know each other, but also the trainees get to know each other. And that's how we build that international community. So that's the idea that we all believe in, that synthetic cells is just one example of that international research effort. We think that all of cutting edge research should be international effort. We should not just be putting it behind a paywall or behind a single country's rules and regulations. We should work as an international community. And working as an international community means that we can not only do it together, but then everyone can access the benefits of that work. That's really important because research is concentrated mostly in the global north, and the global south is the frontline communities where the needs for the kind of benefits of those research actually are. And so we have to work as an international community to get that done. I'm hoping we're doing an okay job right now for the last few years building synthetic cells together as an international community. We're hoping to keep doing that. We're hoping to keep growing that community so everyone can not only participate in research, but also benefit from that research. That's the dream, but also the dream that's already happening. That is the reality. Okay, so I want to say thank you for all of you for listening. If you have any questions, you can look me up and send me an email. And last but not least, I want to say really big thanks to everyone in my lab. There is a lot of work that's been done in my lab with the support of our funders, and I want to say big thanks. When I started my lab, I said I'm going to make life from non-living components, life from scratch. That's crazy. People actually joined my lab. They believed in this. So big thanks to them, and big thanks to you for coming and listening. And I will be very happy to take questions, comments, discussions.
Wonderful, thank you. So for those in the space, you can raise a hand and we'll make sure to get a microphone for you. Those online, please feel free to type your question in the chat and we will read them out loud. And in fact, we have our very first online question, so I'll read that out loud now. This is from Francine, and they ask, can you discuss the dangers of releasing these synthetic cells into the environment when there are no natural predators to rein them in?
Yes, and I think I can figure out where that question came from. 2 months ago, we published a science article where we describe dangers of one particular type of synthetic cells that's called mirror life. We described the concerns about releasing of that one particular, so this is just one type of a synthetic cell, and I'll take a step back and talk about the whole field, but now let me focus for a second on that mirror life. We thought about the dangers of releasing a mirror synthetic cell into the environment. And we decided that the benefits of making that particular cell, despite there are a lot of biomedical, biomanufacturing benefits, those benefits are not worth the risks. We did a risk analysis and we decided the risks of releasing that organism would be too high. And some of the risks are if you have no natural predators in the environment, then you can just replicate without anyone stopping you. And so that basically makes an organism that could potentially overwhelm the environment, overwhelm the natural ecosystems. And so what we did once we realized those risks is we decided this should not happen. And so we're calling for a global moratorium on engineering mirror synthetic cells. And so far the response from the international community has been, yeah, maybe you have a point. So hopefully no one will ever make a mirror synthetic cell. There are other types of synthetic cells that are not mirrors, so they use natural stereochemistry, so normal molecules, normal chirality molecules, and those will be recognized by predators. So that's the biggest thing, is you don't have to evolve in the environment for the predators to know to eat you. For example, my dog did not grow up eating bread. I was feeding him from a puppy really healthy food, but the first time he saw bread, he knew it's food and he ate it right away. And that's kind of the concept of all of biology. You don't have to evolve or grow up eating something. You can be introduced to foods at some point and you recognize it as food. And so that's the whole point with synthetic cells made of normal biological molecules. If we release them into the environment, the predators, even if they've never seen it before, they will recognize it as an edible bacteria or bacteria-like organism because it's made from the same molecules. So the danger of being kind of a not coming from these constraints of evolution don't apply to the normal, regular synthetic cells. If they ever happen to be released intentionally or accidentally into the environment, they will be eaten. They will be also susceptible to viruses. So there will be those regulatory mechanisms that will protect it. The question that Francine, I believe, asked is very relevant, though. We need regulations. We, as a synthetic cell community, are trying to develop now regulations on release of those organisms, not unlike the regulations on release of genetically modified organisms, natural genetically modified organisms. Right now, there are no regulations because the field is so young still. But we're getting to the point where those applications are really coming up into the clinic. For example, our collaborator Avi Schroeder have demonstrated that synthetic cells can shrink tumors in mice. So this is the preclinical trial stage already, where we can put a synthetic cell therapeutic into a mouse that's been engineered to have a tumor, and that tumor can be shrunk in a living organism, in a living mammal. So we're getting, a few years away from actually putting them into human patients. And so we need regulations. We need to learn how to really regulate that environment. So a very timely, relevant question, and we as a synthetic cell community are working on developing those policy and regulations.
I believe we have two questions, one back here and there.
Yeah.
Hi there. My name is Junghao Ka. I was just really curious as to all the different applications that you put forth for synthetic cells, whether it be biomedicine, biomanufacturing. I'm wondering which one of these do you think is the most exciting for you and really motivates you in your work to push forward?
Yeah.
Like where do you want your work to progress, in what direction?
To me personally, the biomanufacturing applications are the most exciting. Recently, I got a group of people together into an initiative that we call BioBold, which is intended to build a global network of bioreactors. We're starting modestly with 50 all over the world, bioreactors that use synthetic cell biomanufacturing to produce petrochemicals or compounds that can mimic petrochemicals that then go into the local economy. So that helps us move away from oil and also help us build bioeconomy locally with local communities, not those big centralized billionaire-led companies. And so that's what really fascinates me is this using biotechnology to allow people equal access to opportunity, because that's something really missing right now. Here in a developed country, we have access to those opportunities. We can go work for a biotech. There are a lot of people who have the desire, who have the ability, but cannot access those opportunities. And so that's that part of the application that's really fascinating to me. I have to say that I'm also obligated to be excited about the biomedical applications, because that's what pays for a lot of our work. Both for my lab's work and for the companies that I started, we do develop biomedical applications, and I would love to be able to see that in a patient. This would be kind of an ultimate rewarding experience if I can say the work that came out of my lab can actually cure someone. So hopefully we'll see that happening too. But on the little kids level, it's the astropharmacy. I have to say, sending stuff to space, that's the really coolest part. So, when my sample went to the ISS, I was like, wow, an astronaut's touching my sample. How cool is that? So there's no one answer to your question, and there's a lot of different aspects of it that are fascinating.
Hi, my name is Jackie Ward. I'd like to know, to your previous question about parameters and rules and regulations, what do you anticipate as far as reproduction of these cells? I mean, and rates of reproduction.
Yeah.
How do you manage that before you put it out on the market?
Yes. So for the safety and security of the applications, we actually are happy that they don't reproduce. Because the assumption is that when we start using them as drugs, most of at least the first generation pharmaceuticals based on synthetic cells will not be replicating. And it's a bug when you think about it from the point of view of engineering, like I would love to be able to make a self-replicating life, but it's a feature when it comes to safety. So the first round of applications will probably not replicate. We don't officially have a self-replicating synthetic cell yet. There is very recent work coming out of my lab and another group where we demonstrated that they could undergo one round of self-replication. That paper is about to be published. Hopefully that will kind of stir that conversation and people will start considering that it's actually possible to self-replicate a synthetic cell. The rates of replication are abysmal. Like I get one generation a day, and I so far was able to convincingly demonstrate one generation. I mean, that's ridiculous. We're hoping that one day we'll be able to engineer them to replicate faster. But just to put that in perspective, the minimal mycoplasma cell, so the one that's living cell with a minimal genome, it replicates about once every 20 hours. So a lot of the time, my cultures of that minimal mycoplasma cell mold before the cells get to replicate and cover the plate. So the replication rates will be really, 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 really low. That's not good when you think about doing evolutionary experiments. That makes for a really long PhD if someone has to wait that long for a replication cycle. But it's really good from a biosafety point of view. And I don't see a viable pathway to speeding that up anytime soon because we don't have the complexity in synthetic cells. We don't have the robustness that's needed for a faster replication cycle. So I think the safeguarding will be built into that technology from, at least at first, that it just won't replicate efficiently enough. Once we learn how to replicate them faster, that's probably maybe a decade away. We're hoping that we'll also know that system better and we'll be able to regulate it like the current GMOs are regulated. I realize that wasn't a satisfying answer because we don't actually have like a hard number for replication rates.
Thank you.
Thanks.
You were talking about biomanufacturing to replace petroleum projects.
Mm-hmm.
Given that right now we're dealing with climate change from petroleum products and microplastics, is there a way to manufacture a replacement that would not just simply continue those two issues?
Yes, that's kind of built into the whole concept of biomanufacturing is, we talk about it as replacing petrochemicals. That's kind of a shortcut. Right now, the efforts are on making compounds that will functionally replace petrochemicals, but will be biodegradable. And that's obviously kind of a tricky balance that you have to pull. There are people engineering bacteria that can break down plastics. The problem with that, though, is that the whole world is made of plastics right now. So if you build a bacteria that eats plastics too efficiently, you end up with basically an Andromeda strain. You will end up with something that will just go on and eat not just the microplastics that you want to degrade, but also all the plastics that you're using right now, and that obviously wouldn't be good. So that's a tricky problem, but yes, when we talk about making replacements for petrochemicals, we have the design constraint that is, let's make it actually eventually biodegradable. I don't know what chemistries would that be yet because that's still being developed, but that's kind of the hope and dream of our field is that there will be plastics that are not as persistent in the environment.
And we have our last question coming from online. This is from Jeremy. What are the key methodological steps and workflow considerations in designing and constructing personalized synthetic cells for specific applications, e.g. regarding the building of the genome?
So that's where the workflow is changing as we speak. As of very recently, I would tell you that you need to sequence the patient. You need to then do in vitro studies that will tell you what exactly is the molecule that you need. For example, if it's a cancer, what is the receptor on the surface of a synthetic cell that you need in order to target that cancer. It's changing right now because of the AI, the rise of the predictive AI that helps us figure out how molecules will behave without the actual need to do a lot of those kind of experiments on the ground is changing that workflow. And that means that hopefully soon we'll be able to sequence, again, you have to sequence the patient, although these days it takes a day. You have to sequence the patient, figure out what mutant, what variant, again, using cancer as an example, what variant of a tumor that is, and then you can use your library of known interactions of the synthetic cell receptors with different receptors on the surface of the cancer cells to design that protein, synthesize the DNA, and then build a synthetic cell. So hopefully that kind of a life cycle will be significantly shortened. And we have some very preliminary data that show that this is possible.
Wonderful. So I think that's all the time that we have.
Thank
you so much. This has been a fantastic presentation. For those of you online, we're going to be closing up the live stream, but thank you for your contributions and all of you here as well. Just a reminder that this conversation and presentation is already available on our YouTube channel if you'd like to revisit and share it. So have a wonderful rest of your day. Enjoy your Friday, and we hope to see you again soon, everyone. Thank you so much.